Hello there. Today in this class, let us understand about molecular biology paper, Central Dogma. So what is Central Dogma? The Central Dogma of molecular biology describes the fundamental flow of genetic information within a biological system. And this Central Dogma was first proposed by Francis Crick in the year 1958. It is the most common and simplified form. It states that the flow of genetic information is primarily unidirectional, that is DNA to RNA, RNA to proteins. Or you can also state this as the central dogma of molecular biology is a theory stating that genetic information flows only in one direction, that is from DNA to RNA, RNA to protein. That is, this is DNA, then RNA and protein. And further later, once they discovered the reverse transcriptase enzyme in some of the RNA viruses, that is like a retroviruses like HIV, that could synthesize DNA from an RNA template using an enzyme called reverse transcriptase. That allowed that to integrate the genetic material into the host cell's DNA, right? And later that was bidirectional. First RNA to DNA, then DNA to RNA, and then further about proteins. So this is about the central dogma. So here, what is transcription? Transcription is process of formation of the RNA molecule, is known as transcription. What is translation? The process of formation or synthesis of the polypeptide chain. A protein is known as translation. So first, always we'll go through DNA application. DNA can be synthesized by the DNA polymerase enzyme. DNA itself, the application will happen in the presence of DNA polymerase enzyme. And further, RNA synthesis happens in the presence of the RNA polymerase enzyme. Okay, the process of synthesis of the RNA molecule in the presence of RNA polymerase is known as transcription. Then translation, the process of formation or synthesis of the polypeptide chain or protein is known as translation. Here, in the presence of reverse transcriptase enzyme, the RNA molecules which are present, genetic RNA molecules which are present in the some of the viruses that could transcribe in the form of DNA molecule. Then DNA will be going through this particular direction, DNA to RNA, RNA to protein. So this is all about your central dogma. Whereas in the eukaryotes, DNA application happen within the nucleus, whereas in prokaryotes, DNA application happens within the nucleoid, in cytoplasm itself only. Okay, transcription happens within the nucleus in eukaryotes, right? Transcription means the process of formation of the mRNA molecule. Once mRNA is ready, then translation happens in the cytoplasm. Once mRNA is ready, that will be coming to the cytoplasm. In the cytoplasm, mRNA in presence of ribosomes and the tRNA molecule, translation will happen. Okay, so this is the process of central dogma, DNA to RNA, RNA to proteins, right? I hope you understood about the central dogma. Further, to mention about central dogma, the central dogma of molecular biology describes the transfer of information between the nucleic acids and proteins, right? The genome is copied during the cell division by the process of DNA replication in the presence of DNA polymerase enzyme. A variety of RNA products are generated by DNA transcription, one of which is mRNA, which is used to synthesize proteins by mRNA translation. Okay. The majority of the RNA produced by DNA transcription is required for protein synthesis, that is ribosomal RNA and tRNA, then RNA processing, then regulation of gene expression and protein synthesis. So this is all about central dogma. Thank you everyone for watching this video.